graduate program in bioinformatics at UFMG. And it's a great pleasure and honor to be able to receive our speaker today, Professor Zhiping Weng. She's a professor at the University of Massachusetts Medical School and director of the program in bioinformatics and integrative biology. Professor Weng's main research interests are bioinformatics, computational biology, genomics, epigenomics, transcriptional regulation, molecular recognition, and the research group led by her has developed several widely used softwares in the bioinformatics field. Just to name a few, Screen, which is part of the ENCODE consortium, ZDock, a rigid body protein-protein docking method, TEMP, an algorithm for detecting transposable element movement, PyPipes, a small RNA analysis pipeline, FAST, a protein structural alignment tool, among others. In the last year, she was awarded the fellowship of the International Society for Computational Biology, ISCB, for her outstanding contributions to the field. So welcome, Zhiping, and I will give the floor to you.
Thank you so much, Raquel. This is great. Thank you for inviting me, and it's a great pleasure to be here. Today, I'm going to talk about our protein docking work upon Miguel's suggestion to begin with. So my lab does quite a few diverse research areas, including protein docking and genomics, epigenomics, as you just heard from Raquel. And I would be glad to speak about another topic in a month of time. So today, I'm going to talk about ZDock and some of our recent developments of protein-protein complex structure prediction. This project has been in my lab for quite a long time, I would say 2 decades. So it was quite fun for me to review everything and go over what we have done and give you a summary point of view today. Actually, this area in my lab has been a small component in recent years, just because the genomics areas are exploding so quickly. And I have shifted a lot of my effort to genomics. But still, we have been working in ZDock and other related projects throughout. And as you can see, I have some recent results to report to you. So just to put our research in perspective, usually there are quite a lot of experimental tools for studying protein-protein interactions. For example, you can mutate some protein to find out whether or not the mutation affects binding, and you can use East-to-hybrid system to large-scale test whether or not which proteins bind to which proteins. You can use protein microarrays, specifically for solving protein structure. So you can solve 3-dimensional structures of protein-protein complexes using techniques such as crystallography, NMR, and cryo-EM. These are all very, very productive techniques. Nonetheless, all these techniques are still not able to catch up with the large number of protein-protein complexes that the cells utilize. And as a need, protein-protein docking is a computational approach to determine the binding mode of 2 proteins, given the structure of these 2 proteins in an unbound state. So that is the topic of what I want to talk about today, is protein-protein docking. So here is an outline for my talk. I'm going to cover 5 topics here. And the first is I'm going to introduce our algorithm ZDock for protein-protein docking. And then in the second point, I'm going to show you how ZDock performs on a docking benchmark, which we actually maintain for the field, and also Capri, which is a community effort of evaluating docking algorithms. In the third bullet point, I'm going to show you some results of our research for using biological data to improve protein docking results. And the fourth and fifth points are our recent papers that just came out last year. And then this last one is, the fifth one came out in December. And then the fourth one is just in press. It's going to come out soon. So I want to add some new results of what we're doing. Okay, so introduction of protein-protein docking. So if you have the three-dimensional structure of a receptor protein here and a ligand protein here, so can you predict the structure of the complex between these two molecules? So when I say complex, I mean, can you move the two molecules with respect to each other and then come up with a structure that has both molecules in it? So in the simplest construction, this is a problem. If you treat these two proteins as rigid objects, it's a search in a six-dimensional translational rotational space, 3 translational degrees of freedom and 3 rotational degrees of freedom. So 6 in total. And how do you search in this space to find the best interaction that will lead to the most stable binding? However, there are additional complexities. For example, some proteins have flexible parts that will change their conformation upon complex formation. And there are environmental effects such as the pH condition, salt, and there may be cofactors. And so these typically are not taken into account in the calculation. And the last point I want to mention is the scoring functions to measure how strong the binding is are, in theory, really complicated. And what we are doing is to use an approximate scoring function so that we can compute with reasonable amount of time. And as a result, it's not precise enough to reach the correct answer. So these are all the difficulties with the problem. And my lab started to work on protein docking in the early 2003. That's our first paper on ZDock. And I remember Ron Chen was one of my first graduate students. And he was instrumental in releasing the first version of ZDock. So here are the basic assumptions of ZDock. We assume the input structures are rigid. In other words, we do not perform specific conformational searches of residues on the input structure. And we assume we don't have any prior knowledge of the binding site on either of the molecules. And as a result, we scan the entire 6-dimensional space, 3 rotational, 3 translational. And to speed up the search in the rotational space, to speed up the search in a 3 translational space, we use this Fast Fourier Transform Algorithm, which I will explain in the next slide. And then that algorithm is grid-based by necessity. And for the scoring function, we use an atomic atom-based scoring function, which includes a number of components such as electrostatic interaction, desolvation upon binding. And we also have a residue contact potential, which is quite effective in taking into account some other environmental effects. As I mentioned, that the interaction space has 3 rotational degrees of freedom and 3 translational degrees of freedom. And we treat them somewhat differently because we do not speed up the rotational search, and we speed up the translational search using FFT. And we retain 1 translational position for each rotation, as you will see in a minute when I explain the algorithm. So obviously we have known drawbacks for the algorithm. The number 1 is for all docking algorithms, not in particular to us, that of this kind, we need the input protein structures. We don't really fold the proteins to begin with. So instead of sequence, we need the structures to begin with in an unbound form. And then the scoring function needs to be approximate because we use the FFT to speed it up. So there are some constraints in a scoring function. And we don't model seriously like entropy of encounter or any other dynamic effects of the 2 proteins. Unlike some of the more detailed simulations, we don't do any simulations. And as I said, we do not explicitly search for conformational changes upon complex formation, but we do have some built-in effort in a scoring function to kind of soften the molecules so that they're not like so hard. They tolerate some sterical overlap and still yield correct binding scores. So just a bit explanation about this fast Fourier transform algorithm. So for the ligand molecule, here I'm illustrating the algorithm in the 2-dimensional space. We explicitly search in the rotational space. So you can see the ligand is spun a little bit, And then now after each rotational position has been searched, we put it into a grid. So here is a 2-dimensional grid, but in actuality, it is oh, some people are typing on the screen. Can you see? I can see here. So I don't know if we could disable.
Yes, we see.
Some people are getting creative here. So anyway, so we have a grid. We map the molecule on it, and then so we have the receptor molecule here. We don't rotate the receptor because you only need to rotate 1 molecule in order to search for the relative rotational space. So we just do it once for the receptor on the same grid. And then we perform a fast Fourier transform for both the receptor image, discretized image, and the ligand image. And here you see some grid points in red and some grid points in blue. These are different assignments, depends on the shape of the molecule to approximate the scoring function. So the scoring function is a convolution between these 2 grids, and the convolution can be sped up using this fast Fourier transform that cuts down quite a bit of computing time. So instead of explicitly translate 1 grid versus the other, we just need to do 3 FFT calculations. I stands for inverse. So you just need 3 FFT calculations that will give you the correlation of all of the translational displacement. And the highest point indicates the highest correlation, which is the score. And you can see that these high points are like on the edge, okay? That corresponds to 1 molecule being translated along the edge of the other molecule. And at the end, if you pick the translation that leads to the highest correlation, that would give you the complex structure in terms of translational space. So for each of rotation, as I mentioned in the previous slide, we keep one of these relative translation. And then at the end, we have a set of rotations, and then we re-rank their score again. So this FFT transformation can speed up the algorithm by hundreds to thousands fold, which allows us to perform a complete 6-dimensional search without assuming the binding site. Okay, so these colors indicate the surface interior, and then the binding site here is indicated by dark blue. So if that's very abstract in terms of fast Fourier transform, you just need to remember it's an algorithm to speed it up. Effectively, what we're doing is we rotate the ligand molecule, and then we also translate, and then we're just doing an exhaustive search in the 6-dimensional space. And at the end, find the pose that's between these two molecules, the relative position of the two molecules, which will score the highest in terms of our scoring function we define. So if you have any new improvement in the scoring function, you just need to put it into the algorithm, and then you will optimize, and then at the end find the best position to achieve the best scoring function. So our docking program has been around for quite a while, and we also have a server, which is people can upload their protein structures, enter the email, and you can choose additional parameters, and then later on the results will be mailed to you. The algorithm is a bit time consuming still because it's doing a complete 6-dimensional search. So you probably take some time. So here's the average wait time. And if you can see this small text, it shows how many people visited the page and where they are in the past month. So we published this paper that describes this server a while back, and we have another manuscript that's being worked on. We have an upgraded server, which has quite a bit of more functionality to make it more user-friendly, and additional bells and whistles you can adjust to do the calculation. So that's one of the still ongoing work. So with the ZDoc program in the middle, it doesn't give you the correct answer as the top scoring configuration all the time. So maybe for 10 to 15% of the times, you will get the correct answer. So for the rest of the times, you have to do things additional. So you need to rescore. For example, these are the possible things you can do. You can rescore your docking poses using another more detailed function, which is slower to compute. You can perform pruning of the configurations to throw out things that you don't think is correct. You can do all the things that are very fancy downstream using biological information. Like Miguel said, you guys are into network analysis. You can apply your network analysis tools on the configurations found by ZDoc. And you can also do something upstream. For example, if you know some binding sites, you can restrict ZDoc to perform docking on those binding sites. And you can also apply other high throughput data, such as high throughput sequencing, cross-linking, and anything you can use to help guide ZDoc to the correct configuration, which we do quite a bit of that ourselves, as you can see from the examples I'm going to give you. So as one example of binding site, So we have this work with a postdoc who work published in 2014, where you can use the ZDoc output to try to identify interface residues. Even if the docking solutions are not like, even if the number one is not correct, because the docking solutions are enriched in the correct answer, you can use top ZDoc configurations to see which residues are enriched at the binding site, as shown here in red, which can point you to the correct binding site, even if the final complex structure is not predicted precisely. Okay, so that was the introduction of our protein docking algorithm, ZDoc. And now I want to give you some illustration as to how well it works. So I'm going to show the performance on two accounts. One is on a benchmark of docking complexes, and then the other one is on Capri. So first, the benchmark. So benchmark, this benchmark is like we have quite a lot of people working in the field, and when we started our work, we realized that people were testing their algorithms on different complexes. So the results are just not comparable. And we decided that we should make this process more organized. So very early on, we started to publish a paper in docking benchmark. And over the years, we have maintained and upgraded the benchmark and continue to distribute it so that the people would use it. Here is our benchmark version 5. And later on, we also started to collect binding affinities of the protein complexes in the benchmark. So this was affinity benchmark version 2 that was published in 2015. And at the end of today's talk, I will show you that we actually recently added more complexes between antibody and antigen, which are of special interest for people trying to study making, say, antibody drugs. If you have such complexes, it will be very useful for them. So we augmented this benchmark by adding quite a few complexes in that particular category. So for the benchmark, so what makes a benchmark worthwhile? So we have both bound and unbound protein structures for the cases in the benchmark. So that the structures, both the structures and the interactions have been validated experimentally. And we took care to keep the benchmark cases non-redundant in terms of sequence. So you don't see the same complex over and over and bias your algorithm development. And in version 5, there were 230 entries, and we classified them into biological categories such as antibody antigen, enzyme substrate, enzyme inhibitor, and others. And according to how much conformational change upon complex formation, we also classified the cases into rigid, of medium difficulty, or difficult cases. So if you have used our benchmark, you might remember this front page on the web. That was for benchmark version 5, and with the new cases, now we're calling it benchmark 5.5. And if you log on to the site right now, you will see a newer page than this one that includes the additional antibody antigen cases. So you can see that it's actually quite interesting to see how the benchmark is being used by monitoring the citations of our papers describing each benchmark. So you see benchmark 1, benchmark 2, and the one is published in a later year than the other. And you can see that as they accumulate citations, if you normalize the citations by total, you can see when a new benchmark is released, people start to use the new one, and then the citation to the previous one comes down. And then when benchmark 3 was released, benchmark 2 usage, both 2 and 1 usage goes down. So you can see that people are switching on to newer benchmarks. And I want to say that the test cases in older benchmark tend to be included in the newer versions unless we find some reason to remove them. For example, one reason would be if we have a new case that's of higher resolution than the previous one and much better resolution, even though they're redundant from one another, we might swap in a new case. But usually we try to make the benchmark backward compatible so that you don't get surprises when you switch to newer versions. So we continuously evaluate the performance of ZDoc on our benchmark. Here is an example on benchmark 4, when benchmark 4 was developed. When benchmark 5 was developed in the paper, you will see similar figures. I just didn't go and bother to update. So here are different versions of ZDoc. The difference in the version sometimes indicate a difference in scoring function, sometimes indicates a speed up. So let me not go into all the details. And then the 6-degree versus 15-degree indicates the sampling density in the rotational space. So the 6-degree sampling is a denser sampling that will lead to more predictions at the end than the 15-degree sampling. So so if you how do we read this success rate graph? So here the x-axis indicates the number of predictions you allow ZDoc to make. So out of say benchmark 4 has 200 test cases. So if for each test case you only take the top prediction from ZDoc, so this is how successful the program is. It's roughly 10%. It's slightly, I would say, only 1 out of 10 times we get it right. As I mentioned earlier already. So we're not like a slam dunk, so to speak. Here is if you allow the program to make 10 predictions for each test case, what's the chance that 1 of the 10 will be right? So you can see that we're improving to roughly 25%. 10 is quite a reasonable number because for experimentalists, you can look at the 10 and you can further filter to add experimental data, and you can test these predictions and see which one turns out to be true. So out of the myriad of possibilities, you only have 10 to consider. So that is quite a reduction already. So if you allow the program to make more predictions, obviously it will be more successful. And then we also make this plot called the hit count, which is how many of the predictions are hits, means correct predictions. And then you can see that if you have more predictions and you have a higher number of them being correct. And then the different versions of ZDoc improves over time. So 3.0.2 and is the newest one, latest one. It performs better than the earlier versions. Okay, so that is how we evaluate our docking performance on a benchmark. So when we publish the benchmark, we not only evaluate our own programs, ZDoc here, we also evaluate state-of-the-art docking programs, such as 3 of them shown here, SwarmDoc, PyDoc, and HEADoc. And we show their performance. And here are the test cases in the benchmark. And they're labeled by their PDD ID of the complex. So it's easy to identify them unambiguously. And rigid are the categories that don't have conformational change upon binding. And the medium and difficult cases are the ones that change conformation. Okay. So that was our performance on benchmark. So let me continue to introduce Capri. Capri is a community-wide experiment, so to speak, but everybody knows it's a bit like a competition for multiple groups to come together to assess protein-protein structure prediction methods. So roughly for every time, it's like a continuous process. Every time, roughly 50 groups worldwide participate. And it's quite an effort to participate, I have to say. So we have been very active at the beginning, but lately we don't have the manpower because all these targets you need to submit predictions. And started 2 decades ago, and the organizers release a new set of challenges every few months, and with some time limit for submitting predictions. And then there will be an evaluation meeting, and people publish papers every 3 years, roughly. And we have been participating for the whole time, for the 2 decades. So at the beginning, most of the targets have the unbound crystal structures being provided by the organizers. And more recently, they have become much more creative. Now they require making homology models from an input sequence. Sometimes it will be related to protein design. Sometimes it's not protein, but flexible peptides. So it's becoming more and more diverse. So our strategy for Capri has always been using ZDoc as the centerpiece. And as the plot I showed earlier, we surround ZDoc with additional things, such as blocking protein sites based on biological information, if you have such information on which sites could possibly be the binding site. And then after we get the results from ZDoc, we use additional more detailed functions such as ZRank or IRED. Those are also predicted by, developed by our algorithm. And then we cluster our structures to remove redundancy, because for every structure, the organizer will only accept 10 predictions. So we have to pick 10 out of a bunch of top ranking ones to submit. And then we also perform additional analysis. So it's quite a manual work, sometimes involve lots of paper reading. So at the beginning, we were doing really well. So here are the results for the first evaluation method. And we were doing so well that at some point, ZDoc was run by all these other participants as well as the first step to form their entry structures. So this is, I was just looking up the last evaluation that was published last year. We were part of this effort still, even though we didn't have time to submit a prediction for every target. We're somewhere here. Okay, so we are, here are the groups, and they only listed the top groups in this table, and we're somewhere in between. And we published a paper summarizing our performance last year. So due to the lack of manpower, we haven't been submitting. You can see that we didn't submit for a lot of the structures. So, but still, ZDoc is being used by a lot of these other groups to as the first step of generating possible predictions, and then they do additional things to make them more accurate. So, okay, that was our performance, and I want to give you 2 examples as to how we can improve the results from ZDoc using biological data. And the first example is using cross-linking data. So what do I mean by cross-linking? So here is a paper that describes our effort in 2018 to use cross-linking experiments to improve the results of ZDoc. And here are the complexes in that paper. And you can see they're quite diverse. And some are very interesting looking. A lot of them are actually multimers, meaning like multiple proteins form a complex. And some of them are symmetric, as you can see. This one is a pretty standard one, in my opinion, that one too. And that one is also pretty standard. But these other ones are quite exotic looking. So here is a histogram indicating the number of crosslinks you can get for each of the complexes. And then among the ones we show here, each complex gets 1 to 7 crosslinks. Okay, and what do we mean by crosslinks? So cross-linking is a in vivo technique. You throw this thing into the cell so that they can cross-link between lysine residues, any kind of lysine residues. Sometimes it will be internally to one protein structure, and the ones that are useful for us would be between a receptor and a ligand. So once you have this thing, so this thing looks like this actually, and has a group like this thing is the same as this thing, and then it will cross-link 2 lysines. Here are shown a blue lysine and a red lysine, and then you can pull it out. It has a reporter inside, and then the mass spectrometry will reveal what this is. And then you can map what you get back to the protein sequence database to find out which 2 peptides can get ligated together, cross-linked together. So you take the fragments from mass spec, and then you map it back to this UniProt protein sequence database to reveal the residues where they are in which protein. So this provides a relatively low resolution data, but we found it to be quite useful for guiding protein-protein docking. For the JMB paper I just showed you, we took Jim Bruce's X-link DB data. This was actually a joint publication with the Bruce lab. And so we took their cross-linking data and did our analysis. So how do we take into account cross-linking data? So if the mass spec tells us these molecules, they don't look as protein-like as they should be, but proves the point. If one protein molecule is this Pac-Man and the other one is this bullet-looking thing, and if the cross-linking shows this residue in a blue protein cross-links with that residue in the red protein, okay, we have a distance cutoff for this cross-link because it can only reach a certain distance. So if you have a docking result, for example, like this one, and if the distance is shorter than the cutoff, then we say the docking result is consistent with the cross-linking data. And this distance is even shorter, so obviously that's consistent. And that distance is really long and not consistent, so we would reject these docking results. So that's how we use cross-linking data to filter our docking results. So instead of using a Euclidean distance, which is not physically rigorous, we can also use this void volume distance using an algorithm called the XWalk. This is the reference to XWalk. And and it walks around. It doesn't penetrate the protein structures. Okay, so with the XWalk, this one is also too long for the linker, so we will also reject this one, but this will be the correct one at the end. So so if we do ZDoc without constraints, okay, for these many number of predictions we made, here are the test cases that we can succeed among the data set we tested on. But with these different kinds of constraints, different ways of incorporating the constraints, you can see the number of cases we succeed roughly doubles, depends on the cutoff you use. So that is the take-home message. So you can use cross-linking data to roughly double the correct predictions because you throw out the ones that don't agree with the cross-linking data. As I hinted earlier, that there are some complications with the cross-linking data when you have a multimeric complex. So here, these two proteins are the same. They just colored slightly differently. So you have two of the red molecules binding to two of the blue molecules. And then the two red-blue complexes form a dimer. Okay, so this blue is the same as this blue in terms of sequence. And that red is the same as that red in terms of sequence. So this is just another example with a higher symmetry. So when you have a complex with many copies of the same protein, but the interfaces is distinct, the crosslinks will be coming from all of these distinct interfaces. And you can't tell whether or not they're associated with the same or with different interfaces. So it's quite a complication. As you can see from the panel of structures I showed you, these kind of multimeric complexes is quite prevalent. So in the paper, in the GMV paper, we actually came up with a computational method to deconvolve these kind of cross-linking data using protein docking. So we will start with our component proteins, and we will find out a matrix, compute a matrix as to what kind of interface these cross-links correspond to. Okay, and then we actually perform the docking, focusing on interface 1, interface 2, and then we will build the multimer to see if at the end they agree. So so for example, like this one, at the end we realize this kind of structure will not agree with the cross-linking data. And then this one would agree, and this one would also agree. So at the end, we can figure out what kind of multimer the complex should be. So in summary, we can group cross-links by interface, and then we perform pairwise docking, and then we symmetrize the structures. And then as a result, we can figure out the similarity of the symmetrized interface, and then which ones is the correct stoichiometry. Okay, so that hopefully gives you an idea as to how we can use biological information to help improve the performance of ZDoc. So next, another effort is to use template-based predictions to augment ZDoc performance, which is protein docking-based predictions. So what do I mean by template-based predictions? So it's a bit like homology modeling for modeling protein structures, just one protein structure. You find a template. So here we're interested in predicting protein complex structures. So we need to find a template for a complex. So since we're doing template-based structural modeling, we sometimes may as well start with the sequences. So now instead of using two structures, one for the receptor, one for the ligand, we use a sequence we start with the sequence of the receptor and then the sequence of the ligand. And we go to a monomeric template library. So this is a library of monomeric proteins. And to find out whether or not we can model the homology structures, homology modeling of the receptor molecule and the ligand molecule. Okay, so we find the best matching structure of the participating proteins from a database. And then we go to another library. Now, instead of having monomeric structures, now this library has complex structures. And then we're trying to find out if there is a match for our two proteins. And if there is, we can use structure similarity of the whole structure. Is there any similarity between this structure and that structure? Or we can use interface similarity, only the residues at the interface of the complex template. And then we can fish out a template, and then we just homology model, superpose this structure onto that structure, and then superpose the blue structure onto the template complex. And at the end, this is our prediction. So it's not docking at all, but it's like homology modeling, and you just superpose using structure orientation. So template-based approaches have become more and more useful now that the databases are being more and more complete with protein structure for complexes. So we did this analysis and see how about comparing a template-based method. We chose one called the Spring, just because that's one of the best ones, but there are other ones available as well. We happen to use this one. And then also on ZDoc. And what we found was the two approaches are actually quite complementary. In particular, the template-based methods work really well if you have a high sequence similarity template already in your database. Then you kind of know the answer, and the method works really well. And then the other one, on the other hand, ZDoc works well when there is a minimum binding induced structure changes. And it particularly wins when the interface is novel and you don't have any template for the interface, and ZDoc can predict it using structure information. So they're very complementary. And this one doesn't care about conformational change because it just superposes. Okay, so we thought about developing an automated way of choosing between Spring and ZDoc predictions. So here is a paper. I think I forgot to put the reference. So this is a paper we have already published in 2019 in bioinformatics that combines Spring and ZDoc. And my postdoc, Sweta, came up with a name called a Zing, just a hybrid of these two words that combines the predictions of the two approaches using uniformly, we re-rank all the predictions using a uniform ranking score. And in this success rate plot, which you have seen by now, you can see that the ZDoc performance is a bit like what I showed before. And then the Spring performance for these test cases, because they have quite a bit of template available, it's actually better than ZDoc if you just compare them off the bat. But if you combine them using our method Zing, it actually performs better for both of them. And we have a way of ranking them so that the sum is better than the addition of the two parts. Okay, so that's another approach of combining additional data to improve ZDoc performance. So now I'm going to go on and just give you a brief overview of our two latest papers. One is we want to augment our benchmark with additional complexes, which are specifically in the category of antibody-antigen interaction. So this is a paper that's in press that is going to come out soon. So I put a date of 2020, and we have additional cases, and then we give examples as to what they can be useful for. Some of the additional cases are structure only. Some have binding affinities as well. And we show additional applications. And this is a joint work with Brian Pierce, who was a graduate student in my lab, but now he is an independent professor at University of Maryland. So here is a bar chart to show how many new cases we're adding. So benchmark 5 is here. And then when you add new cases, it's here. So the addition is quite significant. The new ones are even more than the existing ones in that particular antibody-antigen category. And then they roughly show similar distribution in terms of different kinds of antibodies. It's kind of interesting. So there are, you see, human or humanized antibodies, these cyan bars. And then these are mouse antibodies, rodent antibodies. And then the purple are these single domain antibodies, such as made by camels. Camels are a kind of animal, one of those animals that actually produce, in addition to the typical antibody that has a heavy chain, has a light chain, and they produce a single domain antibody. And they are very convenient for pharmaceutical purposes because they have only one domain and they fold, purify much more easily. So you can see that our new additions have a dramatic improvement in terms of including this kind of single domain antibodies. And the new addition has more cases that are difficult or medianly difficult in terms of docking. And here shows some specificity. Here's an example of a single domain antibody at the bottom. This one is the antigen. And you can see between bound and unbound, this loop is like flopping tremendously from one position to the other. Here is a comparison between a single domain versus a typical two-domain antibody. And you can see the CDR3 loop even form a hairpin. That's kind of crazy. And here is the example of the flu virus. This is influenza's coat protein in the middle. And then our cases, there are so many different cases of antibodies recognizing the same antigen. All of these are antibodies. And I don't want to go into details here, just to give you an idea. Like when we have a new benchmark, we will evaluate the performance of docking algorithms, like including ZDoc, our own, and these other algorithms. They perform as varying. And if you are interested, you can read more in the paper about details. And also for roughly half of the cases, we have binding affinity available. So there are a whole bunch of algorithms that can predict the binding affinities of complexes, such as specifically the delta G, binding free energy. So we evaluate a bunch of predictions, including our own. We have our own, like ZAP is one of our algorithms, Z-ranks, these are our own. I think there is another one. Okay, I don't see it here. But at the end, a scoring function called the Rev15, which is affiliated with the Rosetta algorithm by Dave Baker's lab. It's somewhat better than the other methods. And so I thought I added another slide on how well it does. But anyway, so that's the new paper on the new antibody antigen cases and the kind of things we perform to take advantage of the benchmark. So the last few minutes, I want to mention another project. Obviously, we're particularly interested in antigen recognition. So antibodies are the molecules made by B cells. There are also T cells, another myeloid cells, white blood cells in your body that produces T cell receptors. They also recognize antigen. So my lab has been studying T cell receptors for many years. So this is one of our recent papers. This paper just came out December 2020, and it's done by my student, Tyler Borman. And you can see the usual people here. Brian Pierce, I mentioned. Tom Revlin has been a driving force behind many of the benchmark papers and earlier papers, and also Capri papers. Those were all Tom's effort. And then Brian Baker is a collaborator of ours for many years on T cell receptors. So this project was trying to use high throughput modeling and scoring to try to understand the cross reactivity of peptides bound by T cell receptors. So a little bit of the biology behind the system. So T cell receptor is here. It looks a bit like an antibody, like one arm of an antibody. And then it's a membrane-bound molecule. So T cell will be probably up here. And then T cell receptor sticks out of the membrane of T cell. And it's recognizing specifically a peptide. You can see this purple thing here, being presented by a self, again, membrane-bound molecule called MHC. So actually, MHC is probably the most polymorphic protein in the human population. And the reason that we have very different immunity to different diseases is largely caused by the MHC polymorphism across human populations. So in order for a peptide, specifically a peptide, say from a virus, to be recognized by a T cell receptor, it first needs to be recognized by an MHC molecule, which is our own molecule, to present it like a glove, hold the peptide to be presented to the T cell receptor in order for the T cell to recognize this. Okay, so T cells, their function, the cytotoxic T cells, they target harmful cells, specifically virally infected cells, and then kill them. So that's why they're called cytotoxic. And then the T cell receptor is very important in this recognition. Okay, so this I already explained. It needs to bind to this peptide. It needs to be first presented by MHC. So this problem, we started out, so this is another view of the MHC groove, and then peptide inside, and then this is a T cell receptor. So so this is actually a puzzle, right? So it's estimated that there are maybe 10 to the 8th distinct T cell receptors in a naive T cell pool that our body would ever produce, which is far less than potential peptide diversity, because the peptide is unknown. It's from virus, from cancer cells, from anything you can imagine, which will have a far more diverse sequence space. So the T cells, in some sense, need to be cross-reactive in order to cover the diversity of the peptides. And on the other hand, if you have T cell receptors that are too cross-reactive, it might start to kill self antigens, peptides derived from our own proteins, which would lead to autoimmunity. And there are well-known examples, like multiple sclerosis is an autoimmunity that's T cells killing our own cells. So when Tyler joined my lab, this paper came out by Chris Garcia's lab in Cell, and they devised a technique to use a method to select all the peptides that can be bound by a T cell receptor. So this is a high throughput sequencing based technique. My lab, the other half of my lab is very much into high throughput sequencing. So immediately the paper caught our interest. So we decided we're going to use computational modeling to see whether or not we can predict the cross-reactivity as measured by their high throughput experimental assay, but using a computational approach. So how did we decide to go about doing that? So we, for each of the T cell and then peptide MHC complex, in this whole component, the peptide is different because they fish out the peptide and then they sequence it. The MHC and the T cell receptor are always kept the same for each experiment. And they did perform 4 experiments using 4 pairs of T cell receptor and MHC complexes. But within each experiment, they were varying the peptides. Okay, so we would model all the peptides from their selection rounds. So now, because it's a flexible modeling, so we didn't use ZDoc. Instead, we used the Rosetta program by Dave Baker's lab. And then we didn't model backbone because it would be too computationally expensive. So we fixed the backbone, but we simulated the side chain using the sequence of the peptide. And then we studied a bunch of scoring function. We came up with delta G bind, which is based off Rosetta's protein scoring function, one of the possibilities. And then we scored each structure. And then we see whether or not we can predict their high throughput results. So for example, we start with this template structure for one of the MHC peptide complex. And then this is the peptide inside. And then we modeled it. And this is a modeled structure. And first, in their paper, they actually have some low throughput experimental binding free energy for some of the complexes. We started out to see whether or not we can reproduce that. So this is the experimental delta G, and that is our computational prediction. And you can see we roughly correlate. So the correlation coefficient is 0.7 here. It's quite good. So we were encouraged by these preliminary results. And then we moved on to simulate like hundreds of thousands of peptides in their experiment. Here is a particular T cell receptor MHC complex shown here. And then their experimental technique goes by rounds of selection, 4 rounds of selection. So pre-selection, here is a histogram as to the strong binders here, weak binder on this other end. And you can see when you have additional rounds of selection, and at the end, you see this distribution peaks up, we start to get more and more stronger binding peptides. So the selection is working, and our scoring function and modeling is also working. So here is just more results for additional MHC peptide, MHC T cell receptor complexes. They all show the same performance. So this is the purple one you saw just now, and you can see the peak here for these additional complexes. And we also tested the top 50 peptides that were selected by their experiments for these. This is actually multiple sclerosis peptide I alluded to earlier. It's an autoimmune peptide. And the boxes just indicate which residues are in each of the peptide position, and the black boxes indicate the wild type where we started our modeling. So here are the experimentally selected, and then here is our computationally selected, and you can see the two heat maps agree pretty well with the correlation coefficient of 0.85. And the correlation coefficient gets higher, and then when you go down the rank of the top peptides. So the correlation is still quite high after like the top 500 peptides. So we were very encouraged by these results. We submitted a paper based on these results. And then we got a review. One reviewer was very critical, saying, oh, so why would you do this? Why don't you just look at sequence similarity? Why do you look at structure? Why do you throw out sequence information? So we had to revise the paper. Okay, so the reviewer suggested, can you beat this sequence similarity matrix, the Blossom matrix by Hennikov's? So, okay, we did all the analysis over again, and you can see that here is our structure-based technique. It doesn't beat the Blossom like convincingly, but however, if we combine, again, these two approaches are obviously complementary, because for the structure approach, we don't use any sequence information. And then for the sequence approach, we don't use any structure. So if you combine them by simply averaging the ranking, and you end up with this black curve for your precision recall. Sorry, the recall is clipped off at the bottom. So if you are not familiar with the precision recall, a perfect curve will go from left to right on the top and then go down. Okay, so any curve that's closer to the upper right corner would be a better curve, as indicated by these numbers. So at the end, our results for all four complexes indicate that if we combine our structure approach with a sequence approach like Blossom, we can get an even better approach. So that was the punchline we published at the end. So those are the things I want to tell you today, and hopefully I didn't drag on too much in terms of time. And I want to give you a summary. Protein-protein docking, de novo, coming straight from two protein structures in an unbound state is still a very challenging problem. And as you probably have heard, the DeepMind folks have been making good progress on predicting protein structures. And they have been very interested in moving their force into predicting protein complexes. And when we were working on the antibody antigen test cases, they asked us for the new additional test cases so they can train their deep learning algorithm. So obviously this field requires new additional blood in addition to the typical approach. So I'm glad it's garnering a lot of interest. And if you don't do anything, just do de novo docking, success rate is typically 30% if you allow the algorithm to make 10 predictions. And the post-processing, various post-processing improves results. Success rate can typically be between 50 to 60%. And you can use high throughput experiments to provide constraint to further aid the docking. That will further improve the success rate to above 50 to 60%. However, this data requires a lot of integration. There is no easy way of integrating it bulk in terms of in bulk, and you have to do it for each complex. And we continue to expand our protein docking and binding benchmarks so that we can elevate the level of the field so everybody gets improvement. And then at the end, I show you an example of doing structural modeling, which provide information on the antigen recognition by T cell receptors. And I want to thank the people who did the work. Tom, who has been working on the Capri, a lot of the post-processing, and the bunch of you saw his name, and now he actually left my lab a couple of years ago. He's a senior scientist in a company, Vistera. So Tyler was a graduate student until last December. Now he works in Pac Pharma, and he's the one who did the last project on T cell receptor. Sweta was a postdoc in our lab. She left a couple of years ago to become a research scientist at SUNY Albany. She was the one who did the Weng algorithm, which I forgot to put a reference for. Brian, I mentioned, is a professor at University of Maryland, used to be a student of mine. And then Brian Baker is a professor at Notre Dame University, and he is an immunologist, crystallographer, with the expertise of T cell receptors. And he has been quite a long-term collaboration. We published quite a few papers together, and he was involved in the project with Tyler that I introduced to you today. So that's all I have to say, and I'd be happy to take any questions you might have. Let me just put up the summary in case you have any questions.
Thank you so much, Zhiping. Are there questions? Lisa has one question. I can see here that Ayrton Breno Lisboa, he says, congratulations on your work. I want to ask if it's necessary to run analysis on rigid body docking refinement programs, such as FireDoc, when using ZDoc.
Depends on your protocol. A lot of people first do ZDoc and then take the predictions from ZDoc, and then you can do additional things. Or you can do multiple docking algorithms, such as ZDoc with FireDoc and any other algorithms you might be interested in, and then take the predictions and do clustering and see how they agree with each other, basically pull their predictions together and come up with a smart way of ranking them and choosing from them the ones that you think are more likely to be correct. So there's no need to do one after another. You can do a number of ways, depends on your test case.
Okay, thank you. So Lisa, do you want to open your camera? And
okay.
Yes.
Thank you, Zhiping, to coming and to give this very clear talk. So I have just a curiosity about the scoring function you mentioned. Yes. I would like if you can comment about it regarding, for example, all the Capri competition, for example, what kind of scoring function the best performance tools have? What do you think that can improve the docking tools regarding the scoring functions?
So for if you don't use biological information, I think if you don't use like binding site or all those things additional, if you just do vanilla docking of two structures, like ZDoc's function is, I would say, very competitive. The only one I know of that might be better is for antibody antigen. So if you're particularly interested in antibody antigen,
yes,
yeah, and then I would say Shanda Weider's lab produced this Class Pro algorithm, has a particular, you see, they do better than ZDoc here. Okay, And for antibody antigen, so Z-rank, these other algorithms are not like a full search, I would say. So this one, you see, it's somewhat better here. So if you're antibody antigen, I would say check out ClassPro's antibody antigen specific scoring function. But other than that, you know, I say ZDoc is actually one of the best. That's why people use ZDoc as a default.
So which parameter this method have that ZDoc doesn't have? And for example, which kind of parameter in ZDoc scoring function is are the better weight, or are the, do you understand what I'm talking about?
Yeah, are you asking ClassPro or are you asking ZDoc?
Both. I would like to know if the difference in the scoring function, the difference between the scoring function that make ClassPro in this case a little bit better than ZDoc. For example, ClassPro take in account more the electrostatic potential or the physical chemical parameters, or, you know, which kind of characteristics do you think it's better to predict the interaction? I don't know if there is a general answer for that, but.
So in this case, in terms of ClassPro, if you read their paper, you'll realize what they're doing differently. So as I mentioned earlier, ZDoc has electrostatics, has desolvation, has we call it I phase. We have a paper specifically on that interface contact potential. So I think for ClassPro, the reason it's better is they have a contact potential, which has been specifically trained using antibody antigen complexes.
Okay.
So that's the reason for they are being better.
Okay, thank you.
Yeah, thank you for the insightful questions. Raquel, I can't hear you.
Okay, sorry.
Yes.
Marcela says that thank you for the very nice and impressive presentation. My question is regarding to glycosylation. Several antibody and also antigens as virus present glycosylation in its structures. Can the studies you show handle this data?
Yeah, that is a very good question.
Yes.
For glycosylation, in the test cases in our benchmark, we usually remove glycosylations like het atoms. They show up as het atoms in the PDB structure. We usually remove them. The only way we handle glycosylation is because these sugar groups, we assume, will prevent binding at the site. So if you go to the ZDoc server, there is a way of indicating the glycosylation site, and then the algorithm will block the site from being part of an interface. So that's what we typically do.
Okay, thank you. Adriana Ottoni Ribeiro de Souza says, thank you for the very nice and impressive presentation. And Rafaela Ferreira wants to pose a question. Rafaela, do you want to open your microphone?
Hi, so thank you very much for your talk. Congratulations. I was hoping that we can get some guidelines, some general advice from you. So frequently we'll be interested in protein-protein docking, but we'll have people who are not experts working on this. So when you mentioned that you submit your results, for example, for the Capri competition, sometimes you're going to do some post-processing analysis.
Yes.
So could you tell us how do you decide for which cases you are happy with the original results, or when are you going to decide that you want to do post-processing, and then what are the most frequent post-processing analysis that you do?
Yeah, so I showed this slide earlier. Here is our usual protocol. And we start with rigid body docking using ZDoc. We use the 6-degree sampling so that we can get more predictions to choose. So there are sites, if you don't think the site is going to be in a binding place, we'll block it before the docking, such as glycosylation site we just mentioned. And then after that, we, let me use the laser pointer here. So after we get the structures, we can re-rank them using additional functions such as Z-rank or IRAD. Z-rank was developed by Pierce and IRAD by Tom Beverin. So both of them are developed by my lab, and try to re-rank the structures. And so this is like the kind of things we do, and very test case by test case. And then we actually read the papers about each complex. That's why it's just so time consuming, and it doesn't really reflect the way of the algorithm, because we're not biologists. So sometimes we miss things, and sometimes we misinterpret things. Actually, if you read our paper, so over the years, we have quite a few papers. This one is particularly for Capri, and it has test case by test case, what exactly we did. So you can gain some insight. And then there was an earlier paper in 2017. We have probably 4 or 5 these kind of papers. And if you want to see kind of a protocol, the decisions we made, you can look into those test cases.
Okay, thank you.
Yeah, we're not very successful, I have to say, in terms of biologists. Maybe you guys, we go to the meeting and then we realize, oh, this other group is using ZDoc also, but they did better than we did because they're better at reading biology. Okay.
Okay, thank you.
Thank you.
I think we have a question from Tainá. Raquel, your mic is off.
I'm sorry. Yeah.
Tainá is there.
Go ahead. Go ahead, Tainá.
Hi, Tainá.
Hello, Zhiping. Thank
How
you
are
for
you?
the presentation. It was great. And I
Thank
was
you.
wondering, it's a question regarding biology too. We know that there's not only an interaction between MHC and the T cell receptor during this moment of immunological synapse. Do you think there's a way, or do you envision a way to use the biological data regarding other protein-protein interactions to this scoring function, or to the post-processing after the ZDoc work is done? Because we know that these other interactions are fundamental in say whether the T cell is going to generate an actual immunological response.
Right? Yeah. We haven't done that because it's kind of complicated. So there are additional receptors on the T cells, and then there are additional receptors on the antigen presenting cells. And then, you know, as you mentioned, they form this immunological synapse. And it's like an aggregation of all these molecules all together. Yeah, I mean, we because ZDoc is like a rigid body docking algorithm, and we were not brave enough to go that far away from our comfort zone. So we didn't approach those kind of problems, but I agree, they are very important.
Yes, I ask because I work mostly with things before this interaction. I work with MHC peptide interactions, and I always think, wow, this is nice, but it's so limited because there are all those other things that I'm not taking into account. But
but the MHC peptide interaction is so important now, because that's the first step. We also have some papers on peptide selection on the MHC. If you don't pass the MHC, the T cell will never see it. So I think that problem remains very important now these days with like the neoantigen of cancer cells. I mean, it's a really important problem.
Thanks.
Yes.
Thanks for your questions and answers. Sorry.
Zhiping, give us a spoil. What are you doing with COVID?
Working from home.
No, I mean, are you using ZDoc to study?
I haven't done any structured study. So as you can see from my lab, right now, I'm actually the only person left who is doing structural work. So we didn't have the manpower to move into the let's see if I can get out of here. So I was just going to show you like the people in my lab. We're all genomics, so we haven't done anything useful to alleviate the pandemic at all. So
I have a student docking peptides to some enzymes, to some human enzymes, and they realized that we might try to use ZDoc to actually try to bind the spike protein to those proteins to see if those proteins can add to the spike to cut it, to splice it in the middle.
Okay.
So,
okay.
Right.
Yes.
Yeah,
yes.
we can share the observations, of course.
That will be very interesting. We find that usually other people are better than we are in using our own programs. So that's why we make programs available. Like you can see other people in my lab right now. They're all genomics people.
So you distribute ZDoc, right?
Yes, we do. Yeah.
And did you think to implement with GPU? Maybe it would help to use GPUs?
That is a very good question. We have done ZDoc implementation over time. Let's see. And we had a paper that specifically improved the FFT, went from 3-dimensional FFT to a 1-dimensional FFT library so that we can cut the corners of the grid that were all zero. So in the earlier years, we did improve quite a bit in terms of the algorithm, but we haven't had the manpower again to go from CPU to GPU. And if you have anybody who is interested in that, we can share the code and see if you can get it to be faster.
We can look around, but I don't know any CUDA programmer. But I heard that you can learn CUDA in 2 weeks. What about Raquel? Do you know anyone? She's from the computational department.
No, not actually. Well, myself, I have a question. A different question that I think that might interest to some of our students and colleagues is about your involvement with the innovation ecosystem. I saw that you are co-founder of a startup focused on the development of small molecules. What could you share with us about this experience and this opportunity to contribute to the technological development also?
Yeah, that's a really good question. So I co-founded this company called Argenta a couple of years ago with Simon Xi and Travis Wager, 2 other co-founders. And we're trying to develop small molecules that can target RNA molecules directly, because a lot of proteins are not druggable. Either the surface is too flat or it just doesn't lead to a small molecule drug. So we are starting with RNA molecules to find the compound. And the company is doing quite well. And I'm a founder, so they don't listen to me anyway. So I make suggestions all the time. I'm on the advisory, chairing the scientific advisory board, and I suggest things. But I would say, it's definitely an eye-opening experience to talk to people in the industry, and to try to make what we do more applicable to real life applications. It's really exciting. I think definitely if students are interested in the industry, it's a good way to get involved with a startup at the beginning, because they are very different from big companies. We're just scrambling, trying to do anything possible to make it work. It's quite an exciting and exhausting experience.
I see two very friends in the audience, Ana Benko and Paulo Beirão. If you guys want to appear and say hi. Don't know if you have microphone. Yeah.
Hi, very good talk. I really appreciate.
How are you?
Nice
to meet you.
Very nice talk, and I'm very interested in this issue. I'm going to study more about ZDoc and see if we can use for our purposes.
Okay, thank you.
Yeah, it sounds that you can, if you have partial structures, you can use ZDoc to bind the domains of a single protein.
Mm-hmm. Yeah, it's quite good for that purpose.
Mm-hmm.
Yeah, in fact, we have a proposed structure of very complex structure and peptides that we know biologically that it binds to the original structure.
Mm-hmm.
And we use it mostly to test our proposed structure.
Mm-hmm.
And my former student is also in the audience. And I was really preparing to write to him and stimulate him to have a look at the ZDoc.
That'll be great.
Good. ZDoc. I'll try to install ZDoc. Do you? How many cores do you use? Do you have an idea? Like
yeah,
200 or something.
Or you can just use our server.
No, I know.
Yeah.
Just curious to see how is the demand.
Yeah, I'm in the moment I can't find it, but
we'll start with your server. Sure.
Yeah, start with our server.
Okay.
All right.
Ana Benko says here, congrats also. We are also going to use ZDoc.
See?
Thank you, Ana. And we have a question. We have a question from YouTube. How could we go about automating more the process of selecting the type of post-processing will be used in order to incorporating such on virtual screening methodologies?
Oh, yeah. Yeah. How do you automate it, huh? We have tried. We have a protocol, but it's not really automated because it's really the complex specific. When you have information that's specific for a complex, then it's difficult to automate it. So unless you are studying like the case of the T cell receptor or antibody antigen, maybe you can automate it. But if you are studying different complexes, it's difficult.
Okay. Thank you.
Yeah.
Any more questions? Okay. Okay, Sabrina is also thank you for the great talk. And I also would like to thank you so much. It was very inspiring and insightful, very clear. It was really excellent. And we would like to thank you a lot. And we're going to be here again in the next week.
I hope so. I hope so. Eventually.
Yeah, let's do an advertisement. So then next month, by the end of February, we will have a talk on ENCODE, right?
Yes, I will talk about ENCODE. It'll be a totally different topic. You might be surprised by how different it is.
Yeah, I'm impressed that you deal with two different research subjects, right?
Yeah, I mean, I've been working on both areas for many years, and we also do small RNAs, which is yet another universe.
Great. And you see that in August, you will be having Atima from your department, from your program, talking here too.
Oh, great, great. Asma, yes, she's excellent. She works on RNA splicing and rates of RNA splicing. Yeah, she will give a great talk.
Great. Well, thanks a lot for the while. We see you next month.
Okay.
It is really great to stimulate our students with international talk. I'm sure that they are stimulated by this experience. And the idea is to have one talk per month. So next month we will be with you again.
Miguel, when it's going to be? I said the next week, but it's not in the next week. When it's going to be?
Next month is by the end of February. You can, I don't know from there, Zhiping, but from here, if we put BrazWebinars in Google, we get to our page. You can try, you have the program. Okay. So then we'll have ENCODE. Then, yeah, that's my area. I'd love to see. Okay.
Yeah, just,
the 26th of February.
Yes,
26. I don't think if you Google BrazWebinars from there, you reach, because there are lots of webinars that in the US will take the front. But in Brazil, if we put BrazWebinars, it's the first.
It's the top hit. Okay, top hit. I'll give it a try.
Yeah.
So is it going to be the same link, or you're going to send
it's going to be the same link. Actually, we have the program till December. If we do not have the seminar, we have at least the person who is inviting. For instance, some people from other programs are responsible for inviting the person of that month. I know that they know people from abroad, so it's granted that we will have good talks. Okay. So we'll be having every month an international talk, and we are sharing with all the PhD programs. So then students from Sandro's place, from Nana Benko's place, they are all attending. And so we provide an international seminar every month for all the PhD programs. That's the idea. Yeah.
That's great.
Great.
So very well organized.
It was easy. The idea was to get you talking first, because we were already in a deal for you to come, right? So then we got a few already going, and then some people are inviting other people, right? It's gonna be fun.
It's gonna be fun. All right.
Okay. So I bet that ZDoc is already running for attending our students here. Yeah, and I will get in touch with the idea of trying to use ZDoc to bind the coronavirus spike to some proteins. We could share.
That'll be great.
Interest. Okay.
Okay, thank you so much.
Paulo Beirão.
Bye-bye.
You know, Paulo Beirão is the head of the scientific foundation of our state here, right?
Absolutely. So thank you to the foundation.
We have a great presence here. Okay. All right. Thank you. Bye-bye, everyone.
Thank you.
See you next month. Tchau. Hora de dar tchau, como dizem os Teletubbies.